It is an October morning. The garden is still. The feeder outside your window has been busy for weeks, house sparrows, a pair of goldfinches, the reliable robin that stakes the same branch every dawn. The air has that particular quality of early autumn, cool at the edges, light turning amber by mid-afternoon. You are standing at the window with your coffee when you notice something. The blackcaps are gone. The warblers that have spent all summer threading through your hedges, gone. The swallows that lined your roof tiles like a row of small dark notes, gone too. Not fewer. Gone. And they left without making a sound. Here is what makes this strange, not poetically, but scientifically. Those birds did not simply leave. They navigated. Across hundreds, sometimes thousands of miles, through darkness and weather systems and open sea, without a map anyone has ever found, using a mechanism no laboratory has fully reverse-engineered. This is not metaphor. This is the documented, confirmed, peer-reviewed frontier of ornithology. The papers exist, published in Nature, in PNAS, coming out of the University of Oldenburg, from the Cornell Lab of Ornithology, from the Max Planck Institute for Ornithology. The data is precise. The question that remains is not whether this navigation system exists. It is how, exactly, it works, and that answer is still incomplete. Scientists have spent more than 50 years on this problem. They have made extraordinary progress. And they will tell you plainly, the core mechanism is still not fully decoded. This channel covers exactly this, behaviors that science has confirmed and cannot yet explain. If that is why you are here, subscribe now. And stay for the final section of this video, because there is one detail about how young birds navigate their first migration, birds that have never made the journey before, birds that have never been taught, that will fundamentally change how you look at your garden this autumn. Most people who watch birds assume that migration is learned. That there is something social in it, older birds leading younger ones, routes passed down through flocks the way knowledge passes through families. This is a reasonable assumption. It is the way large mammals do it. Elephants lead calves to water sources. Whales teach routes to their young. For some birds, this is partly true. Young whooper swans and cranes do fly south in family groups on their first migration, following experienced adults who already know the route. But for the majority of migratory birds, the warblers, the flycatchers, the cuckoos, the sandpipers, this is not what happens. A common cuckoo, Cuculus canorus, is raised by foster parents of an entirely different species. It never meets its biological parents. It has never seen another cuckoo. And yet, in autumn, it leaves the British Isles and navigates solo to Central Africa, the same wintering grounds used by its species, the same route, the same timing. In 2020, researchers at the Zoological Institute of the Russian Academy of Sciences confirmed this with displacement experiments. Young cuckoos experimentally moved 1,800 kilometers off their expected route, corrected their path and headed back toward the species' typical migration corridor. First-time migrants. No experience. No guide. What makes this genuinely strange is this: if the route were fully innate, simply burned into the genome as a fixed flight direction and duration, displacement should not produce correction. The bird should just fly in the wrong direction. That it corrects its path implies something more than a simple compass program. It implies something resembling a map. But the mechanism encoding that map in a brain that has never seen the territory it is navigating has not been found. The most widely accepted model for how young birds navigate their first migration is called the clock and compass hypothesis. It was developed in the 1950s and refined over the following decades. The idea is this: a bird hatches with a genetic program specifying a compass direction, say, southwest, and a duration, say, fly for approximately 30 days. When the timer runs out, the bird is statistically likely to find itself within the range appropriate to its species. This model has strong experimental support. Young birds hand-raised in isolation, exposed to no other migrants, still attempt to orient in the correct compass direction when tested in circular orientation cages, devices called Emlyn funnels, named after the Cornell scientist Stephen Emlyn who pioneered their use in the 1960s. The orientation tendency is there, built-in, measurable. But here is the problem the model has always had to accommodate: the world is not a smooth gradient. There are mountain ranges, coastlines, weather patterns, bodies of water that must be crossed or avoided. A direction and a duration alone cannot explain the precision ornithologists actually observe. When satellite tracking became sophisticated enough to follow individual small birds, researchers found that migrants were not simply flying a vector and landing wherever chance placed them. They were correcting, adjusting, making what looked like decisions. Ornithologist Martin Wikelski, director of the Max Planck Institute of Animal Behavior, summarized the gap clearly: young birds have a compass, but not a map. Adults have both. The distinction points to something real: experience genuinely adds precision. But it does not explain how the compass is set in the first place, or what the young bird is reading in the Earth's magnetic field that gives it anything resembling positional information before it has ever made the journey. What makes this genuinely strange is this: a 2025 review published in Frontiers in Physiology by Chernitsov and Utvenko at the St. Petersburg Zoological Institute suggests that first-time migrants may not be following a simple vector at all. They may be navigating by a series of inherited beacons, geomagnetic waypoints encoded genetically that the bird uses to confirm position along the route. If this is correct, then young birds are not flying blind on a heading. They are reading a magnetic landscape they have never experienced but somehow already know. The nature of that encoding, how a wintering ground a bird has never visited leaves a signature in its genome, remains one of the genuinely open questions in migration biology. In the retinas of night migratory songbirds, researchers have found a light-sensitive protein called cryptochrome 4, designated Cry4. This is not a metaphor for sensitivity to the world. It is a molecule. It sits in the cells of the eye, and it reacts to blue light. When blue light strikes it, electrons move within the protein, and in doing so, they form what physicists call radical pairs, pairs of molecules with unpaired electron spins. These electron spins are affected by magnetic fields. The orientation of the Earth's magnetic field subtly changes which proportion of the protein ends up in its activated state versus its resting state. This signal, in principle, would allow the bird to see the magnetic field as a pattern overlaid on its visual field. Not a sense of pull, like a compass needle. Something closer to a visual texture, a brightness gradient that shifts depending on which direction the bird faces and at what latitude it finds itself. The University of Oldenburg, through its collaborative research center Magnetoreception and Navigation in Vertebrates, was the first to measure this magnetic sensitivity in Cry4 extracted from European robins. Their 2021 paper in Nature confirmed that the protein exhibits precisely the quantum effects the theory had predicted, that electrons in Cry4 enter a state called quantum superposition, their spin states entangled, held in that entanglement just long enough for the Earth's magnetic field to influence the chemical outcome. What makes this genuinely strange is this: the question of how a bird translates a subtle chemical shift in the activation state of a retinal protein into a navigational signal that guides it accurately to a location it has never been. The neural pathway from molecule to behavior has not been traced. Researchers know Cry4 is there. They know it is magnetically sensitive. They know that birds whose visual input is disrupted lose their magnetic orientation. But the neural code that converts the signal into directional information, the brain region that reads it, and the mechanism by which it interacts with the star compass and the sun compass and the olfactory landscape, that integration has not been mapped. Consider a single bird. In October 2022, a juvenile bar-tailed godwit, Lamosa laponica, tagged at the Yukon-Kuskokwim Delta in Alaska as a hatchling, took off over the Pacific Ocean. It was approximately 5 months old. It had never made this journey. No adult of its species was confirmed to be traveling with it. It flew, continuously, without landing, for 11 days and 1 hour. When satellite tracking ended, it had covered 13,560 kilometers, from Alaska to Anson's Bay on the northeastern coast of Tasmania, Australia. This is the longest recorded non-stop migration by any animal on Earth. The bird was known only by the number on its tracking tag, 234684. The distance it covered in those 11 days is equivalent to roughly 1/3 of the circumference of the planet. In preparation for the journey, bar-tailed godwits spend 2 months in Alaska consuming protein and fat so intensively that males can nearly double their body mass. Their digestive organs, the gizzard, the intestines, physically shrink before departure to reduce weight, the biological equivalent of jettisoning ballast. Researchers at the U.S. Geological Survey and the Max Planck Institute for Ornithology, who tracked 234,684 as part of a joint project also involving Fudan University and New Zealand's Massey University, noted that the bird passed over multiple tropical islands where it could have stopped but did not. What makes this genuinely strange is this: at 5 months old, this bird had never been to Tasmania. It had no experience of the Pacific crossing. No data on current winds. No knowledge of the route. And yet it arrived. The precision of what it accomplished, threading the correct atmospheric corridors over thousands of miles of open ocean, in a body that had never attempted it before, is what the tracking data shows. What the tracking data does not show is where, exactly, in the genome or the nervous system the map for that flight was stored. There is a question that runs underneath all of this, one that migration researchers return to repeatedly, which is the question of how any of these systems, the quantum compass in the retina, the genetic clock and compass program, the possible geomagnetic beacon map, are calibrated before the first departure. The answer, it turns out, involves the night sky. In 1967, Cornell scientist Stephen Emlen published experiments with indigo buntings, Passerina cyanea, raised in planetariums where the artificial sky could be rotated. He found that young buntings do not learn a fixed star map. Instead, they learn the axis of rotation of the sky, the point around which all the stars appear to turn through the night, and treat that as north. In the northern hemisphere, this corresponds closely to Polaris. But if the planetarium sky was set to rotate around a different star, the buntings oriented using that false pole instead. What they were learning, during a critical developmental window in the weeks before their first migration, was not a constellation pattern. It was a rotational principle. This means that a bird's star compass is not inherited. It is calibrated fresh, each individual, from the sky it happens to observe in the period between fledging and first departure. And researchers believe this star compass calibration is then used to orient the magnetic compass, that the innate magnetic sense is fine-tuned against the learned celestial axis, creating a coherent navigational reference. What makes this genuinely strange is this: the magnetic sense, once calibrated, is used on nights when stars are invisible, overcast skies, deep cloud cover, the kind of conditions that make up a significant fraction of any autumn migration. So the bird is navigating by a magnetic field interpreted through a quantum mechanism in its eye, calibrated against a star compass learned during a developmental window that happens once and never again, combined with a genetic program encoding directions and durations and possibly geomagnetic waypoints it has never personally encountered. And all of these systems must integrate, in real time, in a brain weighing less than 2 grams, to produce accurate navigation across a continent. The integration itself, how these systems talk to each other, which system dominates under which conditions, what the bird does when they conflict, is an active research question with no settled answer. You are back at the window. Same morning, same garden, same amber October light. The feeder is there. The robin is on its branch. But the blackcaps are somewhere over the Mediterranean now, or past it already, threading the thermals over northern Africa. They left in the night, silently, navigating by a magnetic texture in their visual field produced by quantum electron entanglement in a retinal protein, correcting their heading against a star axis they memorized in the weeks after they fledged, following geomagnetic waypoints that may have been written into their genome by ancestors who made this crossing generations before any scientist was watching. The garden has not changed. What has changed is the weight of the silence where those birds were. There is a great deal that science has confirmed about how this works. There is a great deal that science has not yet resolved: the neural map, the integration mechanism, the molecular to behavioral pathway, the precise encoding of a place a bird has never been. This is not a failure of science. It is simply what the edge of a genuine problem looks like. You are standing at it. If you have ever watched a bird in your garden suddenly become restless in a way that felt different from its ordinary movement, pacing the perch, orienting repeatedly in one direction, I would be curious to know exactly what species it was and what month you noticed it. I read every comment.